Good afternoon. Today I'm going to talk about my latest invention, electroplasmonic wireless nanoelectrode uh, array for electrophysiological detection with subcellular resolution. My name is Ahsan Habib. I'm a PhD student in the ECE department. Electrophysiology is the tool to explore electrical activity of electrogenic cells, that means neuron in the brain and cardiomyocyte in the heart. Electrophysiology is the key to understanding in neuroscience and cardiology for several reasons. For example, disease diagnosis, for example, Parkinson's, also to develop brain-computer interfaces for people with disability. However, to achieve this goal, we have to detect a large number of cells. The number is gigantic, 86 billion neurons in brain and 2 billion cardiomyocytes in heart. Now the question is, what is the option we have? One of the options is microelectrode arrays, in short MEAs. However, MEAs have limited, MEAs offer limited number of measurement capability. And this is mainly because they have limited number of this electrode. And this is because of this complex wiring scheme. And this complex wiring scheme is the main reason behind the slow growth of the MEA. If you compare it with the semiconductor industry, although they started approximately at the same time, 1970. If you look at this recent number, a recent CPU has like way more transistor than a recent MEA. To give you more number, I have developed this scaling law. What it tells, like number of electrodes in MEAs get double every 4.7 years. If you consider this rate, then it will take 150 years to develop a device that will detect 86 billion cells simultaneously. So that's long time. We don't want to wait that long. So the question is, what is the solution? Since wiring is the problem, wireless is the very obvious solution. And light is the best wireless solution because light offers unprecedented number of measurements and unprecedented bandwidth. So we can detect whole brain, 86 billions of neurons. However, the major problem of light-based probe is the following. It emits very small amount of light. Because of the tiny cross-section, you can see how tiny cross-section it is, fraction of nanometer. And to overcome this, people use high-intensity light and they get issues like photostability. That means this probe stop emitting light, and they get phototoxicity. That means cell environment gets toxic. Also, signal to noise ratio, SNR, is very small, less than 10. Normally, this number means if it is higher, then the measurement is better, also easier. Since the signal to noise ratio is very small, what people do, they actually do large number of averaging. And that's why they get very low bandwidth. To achieve high bandwidth light-based probe, we have introduced electro-plasmonic wireless nanoelectrode array, where each of these nanoelectrode consists of gold nanoparticle coated with voltage-sensitive polymer. The cool thing about this nanoelectrode is the cross-section. This is the cross-section of the nanoelectrode. This is very high. Even it is higher than the geometrical cross-section of this nanoelectrode because of an electromagnetic effect known as plasmonics. If you compare this cross-section with the conventional probe, you will notice that the cross-section is million-fold higher than the conventional probe. Moreover, our probe has very high sensitivity, extremely high sensitivity, 900% for this much electric field. This high sensitivity and this high cross-section give us a very high signal to noise ratio. With our experimental measurements and theoretical calculation, we have shown we can achieve 50 to 200 signal to noise ratio, or SNR, with our probe. That's very high. We have demonstrated electrophysiological activity detection with our probe. This is our probe. We have millions of nanoelectrode in the active area. This is the cell. We have demonstrated wireless detection from electrogenic cell. This is the signal. We have demonstrated high signal to noise ratio measurement capability with our probe. With this, I'm going to finish my presentation by saying a couple of features about our probe. Our probe is capable of detecting signal from the different spot of this single cell, that means subcellular resolution, and the resolution is only limited by the diffraction limit of light. Also, I told you, like, the cross section is very high if you compare it with the conventional probe. So what we can do, we can use very small, low intensity, my apology, we can use low intensity lights compared to the conventional probe. So we don't get any photostability and phototoxicity issues. Also, signal to noise ratio is very high, so we don't need any kind of averaging. So bandwidth is very high. This high bandwidth and unprecedented number of measurements with our probe give us the hope that it will help us to develop ultra high bandwidth brain computer interfaces for people with disability. I want to acknowledge my mentor, our group members, our collaborators, and the funding agencies, especially UC Santa Cruz, for supporting me through dissertation year fellowship. Thank you. I'll be happy to take your question.